Ectothermic species have body temperatures that tend to track the temperature of the environment around them. The temperatures that ectothermic species can tolerate are of great importance in the context of global climate change. As water bodies warm up, how this will affect where different species live and whether they can cope with a new temperature regime are key to the success of many animal populations. There have been lots of studies undertaken in the lab to look at the tolerance to temperature by ectothermic species, in particular fish. However, the lab is not the real world, and so the usefulness of laboratory-derived tolerance data for predicting climate change impacts is questionable. On the other hand, understanding how an animal's physiology responds to different temperatures could help us predict their responses to climate change. We investigated whether physiological patterns found in laboratory experiments of heating tolerance translate into distribution patterns in natural environments. We compared the physiological responses of fish species in the laboratory to their abundance in the wild by analysing data from 2 published datasets that together comprise more than 800 fish species. For fish in the lab, we calculated their physiological heating tolerance, the difference between the temperature that a fish is acclimated to, TA, and the upper temperature at which it could survive, CT max. For fish in the wild, we calculated population heating tolerance as the difference between the temperature at which their abundance is greatest, T-opt, and the temperature at the warm distribution limit of the species in the wild, T-lim. We found striking similarities between patterns of physiological heating tolerance in the lab and population heating tolerance in the wild, both declining with increasing temperature at similar rates. We also found that, at a given acclimation temperature or optimum temperature, tropical individuals and populations have higher heating tolerances than do temperate individuals and populations. These shared patterns at different biological scales suggest a potentially tight link between physiological and demographic processes and highlight vulnerabilities in fishes from both temperate and tropical regions. Despite these similar, marked declines in heating tolerance at higher temperatures, mean heating tolerances are broadly similar between temperate and tropical fishes at both physiological and population levels. As a result, because they generally experience warmer temperatures, tropical fishes tend to have larger heating tolerance than temperate ones at any given TA or T-opt. For example, a tropical fish population with T-opt of 23 degrees C has roughly double the heating tolerance of a temperate species with the same T-opt. Slopes were not significantly different between tropical and temperate species for physiological heating tolerance, and significantly but only slightly steeper for tropical population heating tolerance. The similar rates of decline in heating tolerance across fish physiology and demography, with distinctive patterns for both tropical and temperate species, implicate a mechanistic link between these 2 biological scales. Whatever those mechanisms are, it is therefore possible that the physiological scaling patterns we see are regulating the range of temperatures that fish species encounter in the wild, in turn driving the abundant scaling patterns we see. One possible explanation for why heating tolerance declines for warmer, acclimated or adapted fishes within either the temperate or tropical clades is thermodynamics. 1 degree C of heating tolerance represents a greater metabolic cost at higher TA or T-opt because metabolism scales exponentially with temperature rather than scaling linearly. The key message from our study is that data from the lab and field on fish thermal tolerances reflect one another, which is good news for physiologists wanting to interpret field patterns and good news for ecologists wishing to understand intrinsic mechanisms explaining their observations of the natural environment. You can read the paper by going to the journal's website. The paper's open access. If then you have questions about the work, feel free to contact the corresponding author, Dr. Nick Payne.